Well, thank you, Hillary, and thank you to the organizers for the opportunity to come present to you today. I'm going to tell you about recent work from the labs of Steve McCarroll and Sabina Beretta on somatic DNA repeat expansion. Our labs work together a lot on Huntington's disease, and I'm going to talk about our joint work on HD, and then I'm also going to talk a bit about somatic instability a little bit more generally. So I just have one financial disclosure. My employer has filed patent applications related to portions of this work. So human genomes harbor more than 1 million loci with simple DNA sequence repeats in which a short DNA sequence, like CAG for example, is repeated a variable number of times. And more than 50 human diseases are already known to be caused by inherited alleles in which these simple sequence repeats have expanded in length. Most, though not all, are primarily diseases of the brain. So one of the more famous of these is Huntington's disease. HD is a fatal genetic disorder. Symptoms typically start in adulthood. They include involuntary movements, but also cognitive symptoms. These symptoms steadily worsen over 10 to 20 years, sometimes more, leading to significant cognitive impairment, complete paralysis, and eventually the inability to speak or to eat. And there's hundreds of thousands of people today living in HD families. And we know that HD pathology involves the death of specific neurons in specific brain areas. And while the early pathology is seen most prominently in the striatum, especially in the caudate, the cortex and the other brain regions are also affected. And there's significant atrophy throughout the brain, as you can see in these MRI images. HD is quite literally the textbook Mendelian genetic disease. 3 decades ago, it became the first autosomal dominant human disease in which the responsible gene was found by genetic mapping. And this discovery helped generate a lot of excitement about sequencing the entire human genome. All Huntington's patients have this expanded trinucleotide repeat in exon 1 of the HTT gene, while most of us inherit a CAG repeat with less than 30 copies of the CAG sequence. Inheriting allele with just even one allele with more than 35 CAGs can lead to HD. And moreover, the length of the inherited allele impacts the age of onset. The longer the allele you inherit, the earlier the age at which symptoms will first appear. And we know that the CAG repeat can mutate somatically in HD patients. Peggy Shelburne's lab showed 20 years ago, using small pool PCR experiments, that DNA isolated from the brains of HD patients contains molecules with a wide range of expanded CAG repeats, in some cases up to 1,000 CAGs. But these observations were rare, they were difficult to replicate, and it couldn't be scaled up. So a series of beautiful genome-wide association studies revealed that genetic variants in other genes, other than HTT, act as modifiers affecting both age of onset and speed of progression in HD. And a really striking feature of these GWAS studies was that many of the associated genes turned out to encode DNA maintenance proteins. And then there have been many functional studies, including ones by Vanessa Wheeler and Ricardo Moro-Pinto and others, that have shown that these DNA maintenance proteins in turn impact the somatic expansions of DNA repeats in vivo. And yet, 30 years after the gene was discovered, mysteries about HD still persisted. What's neurotoxic about those inherited alleles? And why do only some kinds of neurons seem to be sensitive to this toxicity, even though all cells express the gene and the protein? And why is there this long latent period, decades, in which people with the disease-causing allele have no detectable symptoms? Well, we wanted to investigate these questions about HD using single-nucleus RNA sequencing of human postmortem brain tissue. And our lab actually invented droplet-based single-cell sequencing in work led by Evan McCosco when he was postdoc in Steve's lab. But we faced a key challenge. We wanted to understand the impact of somatic expansion of the repeat, but we had no way to measure the length of the DNA repeat at single-cell resolution. So a team in the lab spent several years during the pandemic developing a way to measure the length of that CAG repeat in individual cells alongside each cell's genome-wide RNA expression, and thus its cell type and its biological state. And we do this by making 2 sequencing libraries from each set of bar-coded nuclei. One library is a standard transcriptome library, and the other is what we call an HTT-CAG library, which gives us the sequence of the CAG repeat from the HTT transcripts. And we sequence the HTT-CAG library using long reads. So the barcodes actually serve two functions in the assay. The first is to tie each CAG length measurement back to the originating cell. And the second is that they allow us to make precise CAG length measurements because we can compute a consensus length from many reads, typically hundreds, that all originated from the same source molecule. And this provides very accurate measurements even when the CAG repeat is highly expanded. So with this new technology, we were able to measure somatic instability in HD from human brain tissue in single cells at unprecedented accuracy and unprecedented scale. And we first applied this technology to study HD in the caudate, which is affected early and very severely in HD. And the results were strikingly clear. Although there's a little instability in all cell types, the profound somatic expansion of the CAG repeat is occurring specifically in SPNs, the striatal projection neurons. And these are the vulnerable neurons in the caudate that die in HD. And note that the somatic expansion is both cell type specific and allele specific. It's exhibited only by the disease-causing allele. And this was true in every patient that we studied. The cell type specificity of the disease matched the cell type specificity of somatic expansion. In the SPNs, the somatic expansion was dramatic. We were able to measure some CAG repeats up to 1,000 CAGs. But the distribution of the repeat length observations was strangely skewed. In all donors, the majority of the SPNs had not expanded very much to less than, say, twice the length of the inherited allele. But in every symptomatic donor, there was this long right tail of really long expansions. But they were always just in a small minority of the cells, regardless of disease stage. And we call these armadillo distributions. That's because of the long tail. And we used modeling and simulations to try to work out how these armadillo distributions might arise. And it turns out they can be generated by a model with a few simple properties. First, that the expansion is a stochastic process at the single cell level. It's basically a random walk. And second, that it's a nonlinear process. The longer the repeat gets, the faster it expands. And third, to get that armadillo shape, you need an inflection point in the acceleration, which occurs between about 70 and 90 CAGs. And at this inflection point, the rate dramatically speeds up. And this is what generates the long right tail. So we think of this as two phases of somatic instability. The first phase is slow, with the repeat initially expanding something like 1 CAG per year, something on that order. But after the inflection point, the rate is more than an order of magnitude faster, and then it keeps accelerating from there. At a recent HD conference, actually, two HD researchers, Sarah Tabrizi, who I think is going to be speaking on Wednesday, and Mike Flower, reported that they have established multiple human neuronal cell lines from an HD patient, and they have a spectrum of repeat lengths. And in this system, they're able to actually observe for up to a year the expansion of the repeats in those cell lines. And they've been measuring the rate of expansion. And there's actually very good agreement between what these models predicted, which were done computationally just from looking at the end state, and the kinetics that they're seeing in this model system, which was a really nice sort of emerging experimental validation. And if I didn't say so, this is unpublished data, but Sarah said it was okay if I told you about it. And one of the key questions we wanted to understand in our striatum work was how the expanding CAG repeat changes neuronal biology. So this question is not easy to answer, principally because gene expression in an HD brain is severely altered. If you just do a simple case control comparison, there's thousands of differentially expressed genes, and there are differentially expressed genes in every cell type throughout the brain. And this makes it hard to isolate any proximal effect of the CAG repeat expansion from all of the downstream sequelae. And this shouldn't be surprising, right? The HD brain has tons of atrophy, cell loss, inflammation, right? It's not surprising that there's like widespread gene dysregulation. But here's the key analytical idea, right? This abundant somatic expansion in the vulnerable cells gives us an allelic series. So we can use this to test whether each cell is specifically responding to its own expanding repeat. And to do this analysis, it's important that we do it within a single donor so that the life history and the disease state are shared, and we do it within a single piece of tissue from a particular part of the brain, and then we must carefully control for cell type. But if you do this carefully controlled experiment, then we can recognize any direct cell-autonomous effects of the CAG repeat length once we've controlled for everything else. So if we do this well-controlled analysis, what would we see if we compared SPNs with, say, 35 to 65 CAGs to SPNs with 65 to 150 CAGs? Well, the surprising answer, shown in this volcano plot, is we don't see any difference. The cells are not responding in these two groups any differently to the length of their repeats. But now, what if we look at the cells that are way out on that right tail? When we do this comparison, then we begin to see hundreds of differentially expressed genes. So here's a more continuous view of this cellular response. On the top is a heat map from one donor, and each row is the expression of a single gene. The columns are our allelic series. These are all the SPNs lined up from left to right in order of their repeat lengths. And in the bottom two panels, we've summarized the gene expression as the median fold change of the upregulated and downregulated genes. And you can see that the expression is quite stable until a cell reaches about 150 CAGs near the right-hand side of the plot. The cells aren't responding transcriptionally until the repeat reaches this threshold. But then a transcriptional response starts, and it intensifies as the repeat expands further. And we found that this result was highly reproducible in all 6 donors we analyzed. There's a consistent threshold of around 150 CAGs at which a consistent set of genes begins to become dysregulated in the SPNs as the cells respond to the expansion of their own CAG repeat. And this effect intensifies as the repeat gets longer. This was a really surprising result and nothing we would have expected. We called this period of gene dysregulation phase C. And there's one other important thing we learned from these early experiments. In addition to the progressive phase C changes, we also noticed that in a subset of the cells with the longest repeats, there was derepression of many genes that you wouldn't, definitely should not, see being expressed in healthy mature neurons. These include, among others, regulators of embryonic development and genes like CDKN2A and 2B, which are associated with senescence and cell cycle arrest. We called this phase D the derepression crisis. And this cellular response was seen mostly in cells with very long repeats, but it didn't appear to have a specific threshold. Instead, the probability of a cell being in phase D tended to increase as the repeat got longer. So our expectation before these experiments was that we might see a toxic mutant Huntington gene that was becoming gradually more toxic all during the length of a patient's lifetime. We didn't expect to see such a delayed cell-autonomous response. But what these experiments suggested and what they eventually led us to was what we call the ticking DNA clock model. So in this model, for each neuron, there are 4 phases. In the first 2 phases, the CAG repeat in the cell is expanding, slowly at first, about 1 CAG per year, but also accelerating as the repeat gets longer. And then when the repeat gets to about 70 to 90 CAGs, the expansion rapidly speeds up in phase B. Once the repeat length reaches about 150 CAGs, the repeat becomes toxic to the cell, as evidenced by the phase C gene dysregulation. The effect is subtle at first, but the toxicity increases with the repeat length. Then at some point, the cell experiences the derepression crisis, phase D, and is then lost. So a really important characteristic of this model is that while each neuron goes through the 4 phases sequentially, each neuron does this independently and asynchronously. And then the disease phenotype, the symptoms, emerge only when sufficient numbers of neurons have reached the toxic phases. So let's return to the questions that we started with. What's neurotoxic about inherited HD-causing alleles? Perhaps nothing. The data suggests that they may not be toxic until the repeat expands. And why does neurodegeneration take decades? We actually think it doesn't. What takes decades is for the neuron to reach 150 or more repeat units, but once neurodegeneration starts in a given neuron, it's a relatively fast process. And why is neurodegeneration so cell type specific? Well, that's just because somatic expansion rates vary by cell type. We now think that the pathogenesis of Huntington's is a DNA process, not a cell biological toxicity process, a DNA process for almost all of a neuron's life. So the preceding work is from a paper we published over a year ago now about HD in the striatum. And since then, we've been working to understand how well this model does, or does not, generalize to other brain regions, and especially the cerebral cortex. Excuse me for just a minute here. So cortical and striatal neurodegeneration in HD appear to be fairly different. Different types of neurons are affected, and while most striatal neurons are lost, and lost early, cortical neurons are lost later in disease and in smaller numbers. And the observed gene expression changes in the different parts of the brain are really pretty different. And so this has led to hypotheses that the underlying mechanisms may be different. And in particular, an idea that maybe the striatal pathology might somehow be causing the cortical pathology. And this is an important question, right? Because if this was true, then it might be a reasonable idea that you could treat the striatum and the cortex would just take care of itself. But we don't think this model is true. So to address these questions, we've generated new data from multiple brain donors in multiple cortical regions. In total, about 15 times the data in our earlier striatum work. And in the cortex, we found familiar patterns. First, there's abundant somatic expansion in the vulnerable cell types. As in the caudate, the observed repeat lengths exhibit these armadillo distributions, indicative of 2 phases of somatic expansion, and there appears to be a similar threshold for the transition from slow to fast expansion. The long expansions are predominantly in the pyramidal neurons, which are the most vulnerable cells in the cortex. We also observe phase C gene expression changes in the cortex. Remarkably, as in the striatum, the phase C changes commence when the repeat in each neuron reaches a threshold of around 150 CAGs. There's an interesting twist, though, which involves the specific gene expression changes that appear during phase C. And before we get into those specific changes, I want to look at the consistency of phase C within the cortex. So we found that the phase C changes are very much the same in the deep layer versus the upper layer neurons. We also found that they were very consistent region to region, here comparing motor cortex and cingulate cortex. But if you take any of the cortical phase C changes and compare them to the striatum, they're completely different. So while phase C has the same CAG length threshold in cortical and striatal neurons, the cell autonomous response involves very different genes. But there is an apparent underlying theme to these gene expression changes. In the caudate, we had noted that the phase C minus genes, these are the ones that are going down in phase C, were enriched for markers of SPN cell identity. And we sometimes describe the net effect of phase C changes as a kind of erosion of that specific cell identity. And we see that the same sort of thing is happening in the cortex. The C minus genes in the cortex are enriched for markers of specific types of pyramidal neurons. So the theme of identity erosion appears to be the same, although the specific genes involved are quite different because these are very different types of neurons. And there are multiple different subtypes of pyramidal neurons. But the interesting thing is that this pattern is seen in all of the different subtypes, regardless of their detailed identity, cortical layer, or connectivity. The clear differences we see, though, is that between the neuronal subtypes is how many of them have reached the phase C threshold at any given time. And maybe you can guess why. The various subtypes of pyramidal neurons have wildly different rates of somatic expansion. How different? Up to 15-fold variation in the amount of expansion they undergo during a human lifetime. The estimates of the somatic expansion rate for each pyramidal subtype are highly reproducible across cortical regions, as you can see here. This is data from one donor across 12 different cortical regions, where we're plotting the repeat length distributions of each different neuronal subtype. And this cell type specific propensity for somatic instability is also reproducible across different donors. We're showing this here in both motor cortex and in cingulate cortex. And the rates of somatic expansion of the different subtypes of pyramidal neurons, they predict their vulnerability, their rate of cell loss in HD. So we conclude that somatic expansion, not cortical area, not connectivity, appears to be the driver of neurodegeneration. We also observed cells undergoing de-repression crisis, the phase D pathology in the cortex, as we did in the striatum. Phase D in the cortex involves many of the same genes as phase D in the striatum. As in the striatum, the de-repression appears to be abrupt and mainly affects cells with the longest repeats. So while the phase C response involved many different genes in cortex compared to striatum, the de-repression crisis appears to be a very similar process. So to summarize this part of my talk, what we found in the cortex was that the cellular pathology follows the same phases and the same thresholds as we saw in the striatum. The key differences were the following. First, the kinetics of the somatic expansion varies greatly between different types of neurons. And second, the hundreds of genes dysregulated in phase C are also cell type specific, although perhaps with a common underlying theme. In contrast, the de-repression crisis looked similar across brain areas and neuronal types. So now I'm going to switch gears a bit, and I'm going to talk about some work studying somatic instability in large biobanks, specifically UK Biobank and all of us. And this was a wonderful collaboration with Margot Huyuel and Poru Lo. Margot was a postdoc in Poru's lab at the time, and she's now started her own lab at UCLA. I've had the pleasure of collaborating with the Lo lab on several projects over the last few years, and it's been great fun. So this work involved in part a really audacious goal, and that was to see if we could detect somatic repeat instability in large whole genome sequencing data sets. And if so, what could we learn from studying it in 900,000 people? So if you have any experience with short-read sequencing analysis, you may know that just simply measuring short tandem repeats in the germline is a pretty challenging problem, much less trying to robustly measure somatic mosaicism from short-read data. So Margot had to solve some really hard problems to do this. First, she had to refine some existing techniques, like those used in Expansion Hunter. And then she's developed some new approaches, including like this method to computationally correct for the PCR stutter that happens during bridge amplification on Illumina sequencers. And she had to optimize everything so it was practical to run it at biobank scale. But after all this work, the end result was that we were able to detect measurable somatic instability at 20 different DNA repeat loci. We saw somatic instability at several known disease loci and instability in repeats with many different motifs, not just CAGs. And of course, this is almost certainly an under ascertainment just because of how hard it is to pull this signal out of the data in the first place. So we found that many long somatically unstable repeats are surprisingly common. In fact, most of us have at least one and likely multiple unstable DNA repeats in our genome that are expanding over our lifetimes. But you may be wondering, how did we know that what we're measuring here was really somatic mosaicism and not like sequencing artifact? Well, there are two compelling lines of evidence. And the first is that while somatic instability generally increases with allele length, which would also be true probably for sequencing artifacts, we could see that the somatic instability was also increasing with donor age when you control for allele length, suggesting that it's a process that's going on throughout life. And the second line of evidence, which was in many cases more powerful, was whether the repeat instability was associated with known modifier alleles. So I'm going to digress and spend a moment to introduce these modifier alleles. I want to talk about what they are and why they're important. So in the HD field, there is what I'm going to call the consensus view of the underlying mechanism for somatic expansion, at least in post-mitotic cells like we see in HD. So it's based on the idea that if you have a long, repetitive sequence of DNA, that whenever the two strands separate and reanneal, for example, during transcription, there's a chance that they might reanneal out of register. And if they do that, they create what's called a slip-out structure, as illustrated here, where there are these little bubbles of mispaired DNA. And those slip-out structures would create conformational kinks in the double helix. And then those kinks will be recognized by various DNA maintenance complexes. And those maintenance complexes will then come in and they'll cut one strand of the DNA and they'll excise it and they'll resynthesize it from the other strand, resulting in length mutations, usually expansions. So the somatic instability modifier alleles are generally genetic variants in the genes involved in these DNA maintenance complexes, specifically any variants that are acting to either accelerate or retard the rate of somatic mutation. But because so why do we care about modifier alleles? Modifier alleles are of great interest for several reasons. First, they can teach us about the mutational processes underlying the DNA repeat instability, like which genes are specifically involved. And second, those genes that are acting as modifiers are potential therapeutic targets in HD and perhaps for other diseases. But for the development of such therapeutics, it's important to know how the modifier is acting in different contexts. For example, at different loci or in different tissues. So because we're working in these large biobanks, we could use our measurement of somatic repeat instability as a phenotype. And we could run a GWAS and look for associations to these genetic modifiers. So here on top is the result of one such GWAS for the CAG repeat in the TCF4 gene. And when this repeat greatly expands, it increases the risk of Fuchs endothelial corneal dystrophy. So this GWAS identifies many strong associations to known genetic modifiers of repeat instability. We can also compare these associations to results from a similar GWAS for blood instability at the HTT repeat. And this was done in a large disease cohort by the GEM-HD consortium. We see that many of the same modifiers show up, but there are also some interesting differences. So one example is that there's a common modifier allele, a very strong effect in MSH2, and it has a strong effect on many repeats, including the HTT repeat. Yet it appears to have no effect on the expansion of TCF4 in blood. Across the whole study, we found that most of the time, the effects of the modifier genes were generally consistent at different repeats. And that these effects were also generally consistent with what we were seeing in the HD study. These are like 3 examples. And in FAN1, there's actually multiple modifier haplotypes with different effect sizes, and they're shown in color here. And you can see that they're all acting concordantly. But one surprise was MSH3. MSH3 is a strong, statistically significant modifier of instability at TCF4, but it's acting in the opposite direction in blood at the TCF4 repeat than it does at the HTT repeat. Now, to be fair, both of the examples I just used are from the TCF4 repeat. And that's highlighted here in this sort of central column in this table. And what the purple cells are, those are any interactions where the modifier at that repeat is not acting in its consensus direction across the other repeats. So TCF4 turns out to be kind of a weird outlier in this respect, that a number of the modifiers work in the expected direction of effect, but a number don't. But even if you exclude TCF4, we also saw that there was considerable variability in the strength of the effects of the different modifiers on different repeats. And you can sort of get a glimpse of that here in kind of the checkerboard of the green, but it's more clearly shown in the paper in these colored bars on the right of this table. So here, each color is showing the relative effect of 5 different strong modifiers on each repeat locus. And there are some patterns here that make sense. The rows in this table are each repeat, and they're ordered by the length of the repeat motif, from shorter motifs to longer motifs. And based on their roles in DNA mismatch repair, you might hypothesize, for example, that MSH2 and MSH3 would have slightly different effects. MSH2 tends to prefer to repair smaller DNA lesions. MSH3 is usually involved in repairing larger DNA lesions. So you might expect that MSH2, which is in orange, would be more active in the repeats with smaller motifs, and MSH3 in blue might be more active in the repeats with larger motifs. And this is generally true, but there's a lot of variability. And you can also see there's variability in some of the other modifiers, like FAN1 and PMS2. And these locus-specific effects of the modifiers in blood are still not fully understood. But it's also really interesting that the effects of these genetic modifiers can be really strong. So here's an example of a AAAG repeat on chromosome 2. Somatic expansion is associated with allele length here, and this is the sequence of plots going from left to right, showing the length of the inherited allele. It's also associated with the age of the donor. That's the x-axis in each plot. and it's associated with a polygenic score computed from those modifier alleles, and that's shown by the light color through the dark color. And at this locus, those modifier alleles are exerting a fourfold effect on the difference between the slowest and the fastest expanders. So from this biobank project, we learned that there are many somatically unstable DNA repeats in our genomes. And what we were able to observe in this work was undoubtedly just the tip of the iceberg. Not only were there the technical limitations, but rare alleles, especially if deleterious, may be underrepresented in these cohorts. And we only had DNA from blood. There's a lot of evidence that other tissues can have much more instability. We learned that the modifier alleles can have surprisingly strong effects on expansion rates, which might actually make them more attractive therapeutic targets. But we also saw that they can have variable effects at different loci. So we need to understand this clearly if we're going to be developing therapeutics based on them. So in the next few minutes, I want to share with you some brand new work. This is unpublished, but I want to share it with you because I think you may find it interesting. And it all starts with a mystery from the HD world. So most people with HD carry an allele that looks like the top row in this diagram, where the CAG repeat that causes HD is followed by a shorter CCG repeat. But in most people, there's these two synonymous substitutions near the boundary. But in a few percent of people, these synonymous substitutions are lost. These substitutions are called interruptions by the HD field. So when they're lost, these are called loss-of-interruption alleles. And it turns out that people with these loss-of-interruption alleles, they get HD 10 to 12 years earlier than we would otherwise expect. And there's been no clear explanation of why this should be true. It's a really strong effect. So these loss-of-interruption alleles are rare, but we managed to get brain tissue for a few donors with such alleles. And so we wanted to see whether this ticking DNA clock model could provide a framework that might help us investigate, maybe even unravel this mystery. So I'm going to really shorten a lot of work here, but to make a long story short, those loss-of-interruption donors appeared to have the exact same kind of ticking a DNA clock as the other HD donors that have the canonical allele. They have somatic expansion in all the same cell types, both in the caudate and in the cortex. The expansion appears to be slow then fast. They have a similar threshold for phase B. This was one of our leading hypotheses going in. Maybe that threshold would be different. They also have phase C and D pathology. It's in the same genes. It has the same thresholds. All the ways we thought we might see differences, we didn't see any. Although one thing we could see was that the clock seemed to be running a little bit faster. And we saw this in two ways. First of all, we could see, kind of as you might expect, that the vulnerable cell types are being lost earlier. Here, those three dark blue dots are the loss-of-interruption donors, and this is their rate of SPN loss compared to their CAP score, which is a product of their age and their inherited allele length. And you can see that they're losing their SPNs earlier than they otherwise would be if they had a canonical allele. And second, we matched the LOI donors to one or more canonical allele donors, both on their inherited repeat length and their age. We used older donors as the comparators. And we could see here, too, that the somatic instability in the loss-of-interruption donors was ahead of schedule. Okay, so this is good, but we were puzzled at this point and feeling rather unsatisfied. The loss-of-interruption donors are ahead of schedule, but why? How are these two SNPs causing the clock to tick faster? Well, we kept digging through the data, and then we noticed something surprising. In the loss-of-interruption donors, that CCG repeat, which is shown here in blue, it's directly adjacent to the CAG repeat. And in donors with canonical alleles, that CCG repeat is generally really stable. But we saw that there was some instability in that repeat in the loss-of-interruption donors. It wasn't a lot of instability compared to the CAG repeat, but it was there and it was measurable, and it had three interesting properties. First, it was cell type specific. Here I'm showing the SPNs, a vulnerable cell type on the bottom, and all the other caudate cell types on top, and only the vulnerable cell types are exhibiting the instability in the CCG repeat. And second, the CCG repeat instability was haplotype specific. The instability was only happening when the CCG repeat was adjacent to the long CAG repeat on the disease-causing allele. And finally, when we stratified the cells by how expanded their CAG repeat was, we saw that the cells with only modest CAG expansion are mostly stable. In contrast, if we look at the cells with really large CAG expansions, we saw a lot of CCG instability. And in these plots, the CCG length is on the y-axis, so the histograms are kind of tipped on their side. And this looks like a continuous process, a continuous phenomenon. It suggests that the cells that have undergone more CAG repeat mutation have also undergone more CCG repeat mutation. Okay, so what's going on here? As I explained earlier, the consensus view is that somatic instability is precipitated by the formation of slip-out structures in repetitive DNA. Now, most people, at least me, I always imagine these slip-out structures happening within the CAG repeat. And that's probably true most of the time. But what we think was happening here is that in the loss-of-interruption donors, these slip-out structures are sometimes extended and span into the CCG repeat. And this can happen at the energy cost of a one base pair mismatch, as shown by the dot. So why doesn't this happen in canonical allele donors? Those two synonymous substitutions would actually create 5 mispaired bases in the same extended slip-out. So we think what's going on is that those two synonymous substitutions are creating kind of an insulator sequence between the CAG and the CCG repeats that's keeping them from affecting each other. And in the loss-of-interruption donors, the loss of that insulator sequence is creating a larger region of somatic instability, and this allows the clock to tick faster. And I didn't mention, but also this why don't canonical allele donors do this? Because they would have these 5 base pair mismatches. We've actually generated more data recently that suggests that once in a while we think these canonical allele, these longer extended slip-out structures do form in canonical alleles. They just do it at a really, really low rate. It's almost undetectable. So the important take-home message from this, and why I wanted to share this, is that what's going on here is that the sequence context around the repeat in Huntington's is influencing the kinetics of the somatic instability. And while we don't know yet, it's possible that this effect might not be specific to HD. It might turn out that sequence context could affect somatic instability at other loci, perhaps even one you're working on. So if you're looking into somatic instability, if you have a candidate locus, you might want to keep this in mind. So I've told you today about some of the things that we've learned about somatic instability in our labs. First, that it appears to be the key driver of pathology in Huntington's disease in both the striatum and in the cortex. Second, at least in HD, that the kinetics of somatic instability are exquisitely cell type specific. And this also makes them tissue specific. There are loci across the genome that exhibit somatic instability in healthy populations. The modifiers of somatic instability are a powerful way to learn about mechanism, and they're also important in their own right as potential therapeutic targets. And finally, an example from HD showing that it's possible that sequence context might affect the kinetics of somatic repeat instability at certain loci. So I'm privileged to be able to present this work on behalf of an outstanding team of people in the McCarroll and Beretta labs. I want to particularly acknowledge Seva Cashin and Wonsak Lee. Seva was co-first author on the striatum paper, and Seva and Wonsak together have been the principal contributors to the work in the cortex. I want to thank Margot Huyuel and Po-Ru Lo, who led the biobank study. I want to thank our funders, especially the CHDI Foundation, who's been very generous. But most of all, I want to thank the brain donors and their families. Without you, none of this would have been possible. So thank all of you for your attention. I'm happy to take questions.
That was super. Thank you very much. Remember, to those online, please type your questions in the box underneath, and I think Claudia will read them out. And, well, the keen person, why don't we start with a question over there? Yes, and please wait for the microphone. Yeah.
Okay, it's a wonderful talk. I'm just curious about the origin of the expansions. The cells that you're studying, my understanding, are post-mitotic. So it's not like they're getting replicated and then the replication machinery has to go through these repeats. But another possibility is that transcription is disrupting the helix. And so you are getting formation of the slip-outs as a consequence of transcription, and that might provide some explanation for cell-specific effects. Comments about that? Maybe there are things known that I don't know and the rest of the audience doesn't know about the mechanism, but it seems curious that this is happening in post-mitotic cells.
Yeah, great question. So first of all, like pretty much to my understanding, I think to the understanding of the HD field, everything you said is pretty much exactly right. These are post-mitotic cells, like you said, so it's not a replication-based process. It's some post-mitotic process, and that's why we think it is actually when whenever the strands separate and reanneal. And it's less clear. Like there's quite a hypothesis that maybe a lot of it is driven by transcription. But if you try to look and see if it's whether or not HTT is transcribed, it's transcribed in every cell, but only some cells expand. So it doesn't seem like that explains the different rates of somatic instability. And the other thing is that I'm told that DNA can kind of breathe and separate. The helix will separate, like relieve tension and stuff lots of times. So there's I think not a lot of clear evidence to what extent, like if it's true that it's all caused by separating and reannealing, how much of that is transcription and how much is other times that the helix just opens for some other reason. But it doesn't seem like just rate of transcription of HTT is satisfactory to explain like the different rates of somatic instability.
Bob, it's just such beautiful work. As I've told you many times, watching Vanessa and Jim Gasella, traject through all that work, and then you bring it all together, has just been amazing. How do we reconcile the modifier effects that are clearly there and clearly strong, and the modifiers they found for HTT clearly influence many other regions of the genome, with your repeat expansion index that seems to, or with your repeat expansion stochastic process that obviously doesn't depend on changes in the repeat instability genes, given what you're finding in the allelic series. Is there a likelihood that the process is less stochastic in people with risk alleles at modifier effects, or do you think that these things are unrelated to each other? So you're showing a stochastic process by which the alleles expand.
Yeah.
We know that modifiers have a reasonably strong effect. Are any of the genes in your expression profile relating back to the modifier targets, or how do you reconcile those effects with the germline risk factors versus the somatic expansions you're seeing?
I see. I think I see what you're asking. And so is it like whether or not like the expression levels of the modifiers are explaining the different propensities for somatic instability? Yeah. So that's a really good question, and people have tried to look at that. It's not clear that they are. It's not clear that the modifiers vary enough in their expression levels to have that effect. Related story is that the very first time my colleague Seva showed this data, that we have this allelic series of somatic instability propensity across the subtypes of pyramidal neurons, and that it's reproducible, my first reaction was like, this is it. This is the Rosetta Stone that will tell us why there are these cell type specific differences. But despite a lot of work, nothing seems to predict it better than empirically the ordering of the cell types. I think so far, to my knowledge, we've not come up with a better explanation for like why it's so different in different cell types. It just is.
On the following up from his comment, sorry if I missed it, but have you actually looked at the single cell level in people who've got different alleles at these modifier loci to see what's happening, and if the allele is affecting the rate of the somatic expansion, which it presumably is?
Yeah, to some extent. I mean, we don't have like a biobank worth of donors.
Right, yeah.
But yes, we see the predicted differences in the rate of somatic instability of people. And most of the modifiers, the common modifiers, tend to be of relatively small effect at with respect to age of onset.
Yeah, okay.
In HD. It's like a common modifier might have a 1 year,
Right.
effect on age of onset. A strong modifier might have a 7-year age effect, but it's really rare. So it's hard to get a lot of people
Right.
with those modifiers.
Yeah. There's a question right at the top there.
So conceptual in terms of pathogenesis, if I understand correct,
I'm going to have trouble hearing.
conceptual in terms of pathogenesis, if I understand correctly, the neurodegeneration is a quite rapid process, and you have reasonably small number of neurons who are really into this pathogenic stage. Do you expect them to trigger the sort of the second wave of sort of inflammation or something that leads to this rapid progression?
In terms of the rapid progression of disease?
Yeah.
Well, yeah, I think that's basically the model. There's always going to be leading neurons that happen to expand rapidly. Those cells will be lost early. And even though everything is asynchronous, right? Everybody's starting at the same time, sort of on average, there will be a place where many neurons are starting to go into the toxic phases, phases C and phases D. And actually in the striatum paper, we showed that if you just say, well, at what age would people be predicted to have 25% of their SPNs have entered the toxic phase total throughout their lifetime, this is actually a pretty good predictor of disease onset.
Thank you.
Hi, thanks for an amazing talk. My name's Alex. This is quite a speculative question, but you said that the cells with the largest expansions are predisposed to cell death, essentially. Can you learn anything on a single cell level from cells with exceptionally large expansions, why they're protected from dying?
So what was the last part? Whether they're...
Well, if you've got cells with expansions of maybe over 1,000 repeats, let's say, exceptionally large, why are they still? Why have the cells still survived for you to be able to detect those enormous expansions?
Yeah, that's a great question. And I think our best guess is just that they've gotten lucky, right? The repeat is expanding, we think, so fast at that point that there's not a big time window, right? So if there's a little bit of stochasticity at when cells enter phase D, some of them will maybe survive longer. And it's a little bit hard to study because there aren't that many of them. It's very hard, in one tissue sample, there may be only 1% of the nuclei we sample that are in phase D, maybe less than 1%. And we probably also have a little bit of under ascertainment of the very longest lengths. We work really hard to get good ascertainment at long lengths, but we may still be under ascertaining a little bit. So it's a great question, and it'd be really interesting to know if there's differences between those cells.
Well, got another minute. Go ahead.
Thank you. Very amazing talk, and I loved the paper. I was actually teaching it to my medical students earlier this year, and they asked me a question that I could not answer, so now is the time. You basically showed us beautifully that this 150 repeat threshold, and they were saying, so why can't anyone get this? If you don't carry a risk allele in the germline, why are you protected against this? Is this due to all the modifiers, or does it actually happen to anyone?
Oh, so the repeat. So the instability of the repeat is related to how long it currently is, right? That's why it accelerates as the cell goes from 40 to 50 CAGs, then the repeat expansion rate's going faster. If you back that out, by the time you get down to 35, the expansion rate is getting really, really slow. So we think the reason people below 35 CAGs never, or like almost never, if you're born with 35, get Huntington's disease, is that expansion will just never get to 150 in a human lifetime. And there's data from other people that if you do very careful measurements in blood, that even maybe there's a little bit of like somatic instability down to 30 CAGs or something, or even below that. So it's really a quantitative phenotype, but we think the data is suggesting that there's just not a toxic effect till you get to 150. So for a cell population to be like vulnerable, which isn't really a binary state, it's just a question of like how often does that cell type get to 150? And if your expansion rate is slow enough, like many of the cells, they show very little somatic instability. So it's basically just whether you can get to the toxic threshold.
Thank you. That's kind of what I told them, but I didn't know. Thank you.
Super. I think we better wrap up, but that was terrific. Thank you, Bob, and please catch him in the break if you have more questions.